Third would be to explain the second currency, the cells use. Fourth would be to explain the relationship between energy generating as well as energy utilizing pathways. Fifth would be to explain coupling reactions. And then sixth, characterize the three types of pathways. Then seventh would be to explain the modes of regulation of these pathways. And lastly, explain some common organic reaction mechanisms commonly encountered in biochemistry. No? So first, you have the two categories of pathways, mainly your anabolic pathways as well as catabolic pathways. So your anabolism and your catabolism. Now, however, there is a third one which is an amphibolic pathway. No, so you see that. Now, when you say anabolism, these are pathways that would synthesize macromolecules utilizing simple compounds. No, and to do that, the cells need a source of energy, and usually it is ATP. While on the other hand, in catabolism, the cells make use of these macromolecules. They try to degrade them. They try to digest them initially to convert them to their monomeric units. And these subsequently monomeric units would be assimilated and transported to the cells where they will be further oxidized, whereby electrons and protons will be released, and harnessed in the electron transport chain to generate energy. And that's the very reason why we try to degrade or catabolize or oxidize nutrients, to be able to generate that much needed energy in the form of ATP. Now, amphibolic pathways actually refer specifically to your Krebs cycle. You see, Krebs cycle may be used to oxidize. An example would be the product of glycolysis, pyruvate, can be further oxidized, initially converting that to acetyl-CoA. And when it goes through the 8 reactions making up your Krebs cycle, uh, 2 carbons are released, and the electrons and protons released through the oxidation of the substrate or the acetyl-CoA are harnessed into your electron carriers, or what we call coenzymes or reduced electron carriers. And for our Krebs cycle, you have there 3 NADH plus H and 1 FADH2. Okay, so each of these reduced electron carriers will be able to transfer the electrons and protons to your electron transport chain again to generate or synthesize ATP. Now, in other times, this Krebs cycle may be utilized to synthesize, they may be utilized to synthesize amino acids, okay, also other compounds, there. That's why it is amphibolic. And sometimes this is specifically referred to as the final common pathway, especially if you're referring to the degradative nature of the process, because once the macromolecules in the form of carbohydrates, your lipids, your proteins are converted to their monomers or monomeric units, these monomeric units, when further oxidized, will be eventually converted to a molecule, acetyl-CoA, that cannot be characterized as carbohydrate, nor protein, nor lipid. No? Yeah. So this is actually the whole gamut of the different metabolic pathways that you will be studying in biochemistry. To simplify that, there. So you will be dealing with your carbohydrate metabolism, lipid metabolism, protein and amino acid metabolism, there. Energy metabolism, metabolism of lipids, okay. Also metabolism of cofactors and vitamins, and so on. But you can see all of them, especially when you look at the degradative nature of the pathways, will converge to your final common pathway, which is here. Sorry. your Krebs cycle. So let us see the general aspects of anabolic reactions. As I mentioned earlier, they are usually accompanied by use of energy in the form of ATP because the cells need a source of energy to synthesize, no, to create bonds. We know that. While catabolic reactions are usually accompanied by liberation of energy, and that's precisely the purpose of catabolizing or oxidizing nutrients, no, so that the electrons and protons released as a result of the oxidation can be harnessed and be transferred to your electron transport chain, no, whereby ATP is synthesized. Now, reactions may be either endergonic or exergonic. No? There. So, reactions may be needing energy. So, these are reactions that do not occur spontaneously. And other reactions are releasing energy. So, if one utilizes or needs energy and another reaction is the one that liberates energy, we can put them together so that the reaction would proceed. The endergonic reaction would proceed to completion. Now, a good example of that will be the first step in glycolysis. Have you had already a lecture on glycolysis? Wala pa?
Wala pa. Sa Friday pa.
Sa Friday, okay. Now, the first reaction in glycolysis is the first irreversible reaction catalyzed either by hexokinase or glucokinase, no? Whereby glucose is phosphorylated to become glucose 6 phosphate with the help of hydrolysis of ATP to ADP and inorganic phosphate. I will show you the coupling of your endergonic and as well as your exergonic reactions. No? Now, this anabolism as well as catabolism are made up of pathways. Examples of catabolic pathways are your beta oxidation of fatty acids. Glycolysis is a catabolic pathway. Your what else? Other oxidation of your other kinds of fatty acids like your branched chain fatty acids, no, undergoing your alpha oxidation or oxidation of your very long chain fatty acids, no. Then, while anabolic pathways, the example would be your synthesis of glucose through gluconeogenesis or synthesis of fatty acids, no, through fatty acid synthesis. Now, each of these pathways usually has an accompanying very important step known as your, in general, rate limiting step. Or, in particular, when you look at glycolysis, you have there a specific reaction characterized as the committed step. No? And in glycolysis, why is that reaction catalyzed by phosphofructokinase 1 characterized as the committed step? Because later on, you will know that the product of that reaction phosphofructokinase 1, the product of which is fructose 1,6-bisphosphate, that product will serve as an important allosteric stimulator of the last reaction catalyzed by pyruvate kinase, no, to form the product. Because we define the committed step as a step which, when passed, will ensure the formation of the product of the pathway. And in glycolysis, especially in the liver, the product of glycolysis is pyruvate. No? While in other pathways, usually we have a rate limiting step. Now, this rate limiting step may be also named as flux generating steps. Why? Because they try to regulate the movement or channeling of the intermediates of the reactions of intermediates of the pathway to the next or other reactions of the pathway. No? There. Now, this is actually your glycolytic pathway. And highlighted by that red rectangular box, no, is the committed step of the pathway, whereby it's catalyzed by phosphofructokinase 1, whereby your fructose 6 phosphate gets another phosphate and be attached at carbon 1 of fructose to become fructose 1,6 bisphosphate there. Okay. Now, as I've said, that fructose 1,6 bisphosphate will serve now as important allosteric stimulator of the last step catalyzed by pyruvate kinase. Can you see down below? Yes, no? To ensure the formation of the product pyruvate. There. So here is your committed step of glycolysis. No? Now, this is the energetics of the reaction. Synthesis of fructose 1,6 bisphosphate is an endergonic reaction. It needs energy, input of energy. By how much? Positive 16.3 kilojoules per mole. No? While the hydrolysis of ATP to ADP and inorganic phosphate liberates negative 30.5 kilojoules per mole. So if you put them together, definitely the endergonic reaction whereby fructose 6 phosphate is phosphorylated to become fructose 1,6 bisphosphate will become visible. Because if you look at the algebraic sum of the standard free energy of each of the half reactions, there you will still have a difference of how much? Negative, how much? 13.7, 13.2 ba yun? Or 14.2 pala, no? Yes? Tama ba? Hello, class?
Opo, Doc.
Tama, no? May sobra ka pa. Tama, yan. Okay? Now, in our system, you might ask why? What will happen to that extra? Now, our system's reaction are not that very efficient, no? Usually, the extra energy is liberated as heat, no? There. Now, these are the three stages of your catabolic reactions. The first stage, as you can see, as I mentioned earlier, would be the degradation or conversion of your macronutrients into their monomers. So lipids will be converted to their respective fatty acid components, your and glycerol, okay, your polysaccharides or carbohydrates into glucose and other sugars, while your proteins naturally to your amino acids. Now, each of these monomers will eventually be further oxidized into becoming acetyl CoA. No? There. Okay. And that would be your stage 2. And then after formation of acetyl CoA, this will now go into stage 3, whereby acetyl CoA will condense with the most important intermediate of your Krebs or citric acid cycle, which is oxaloacetate, no? Forming citrate, the first intermediate. And that from there, the name citric acid was derived, citric acid cycle. No? This is sometimes also called tricarboxylic acid cycle by the nature of the structure of citrate. It has three carboxylic groups, no? There. And then, as I have mentioned, in your Krebs cycle, there will be generation of your reduced electron carriers in the form of NADH plus H, three of them, one in the isocitrate dehydrogenase reaction, another one in the alpha ketoglutarate dehydrogenase reaction, and the third in the malate dehydrogenase reaction. Another kind, your FADH2, will be generated in your succinate dehydrogenase reaction. Now, each of these reduced electron carriers will be able to transfer their electrons and protons to your electron transport chain. Your NADH may transfer the electrons and protons to complex 1, okay, creating a PO ratio of 3 to 1, no? Or your NADH may be able to transfer their electrons and protons to complex 2, generating a PO ratio of 2 is to 1, no? Have they had a lecture already on bioenergetics and biological oxidation? Did
Yes,
you
yes,
have?
we had,
Meron
we had.
na, no?
Mm.
And I think you did not use 3. Instead, you used 2.5
Yes, saka 1.5 for
and
FAD.
1.5 when the electrons and protons enter by way of complex 2, isn't it? Okay, so you are still using 2.5 and 1.5.
Yeah, we are using those values.
Ah, okay. I hope you know to arrive at 2.5 and 1.5 plus. How do I arrive at that? Now, while electrons are being transferred from one member to the next until finally being accepted by the final electron acceptor, and that is an atom of oxygen, becoming water, being reduced to water, protons are extruded out, no, into your intermembranous space, and eventually to your cytosol, no. What are the areas where protons are extruded out? These are your site 1, which is found in complex 1, site 2, which is found in complex 3, and site 3, which is found in complex 4. In site 1, as well as in site 2, each of these sites will extrude 4 protons each. So that becomes 8. And then another 2, which is extruded at site 3, which is at complex 4, that makes a total of 10. And they found out, class, for this electron transport chain to synthesize 1 mole of ATP, there should be 4 protons that should be transported back inside the matrix. So therefore, 10 divided by 4 would give you 2.5. And when the electrons are transferred or enters your complex 2, then uh, the two sites, site 2 and site 3, will be the ones only involved, no? I hope you're with me, class. That's why 6 divided by 4 will give you 1.5, no? Okay, so there is your oxidative phosphorylation, whereby, as I've said, ATP will be synthesized, no? So here are the 3 stages again of your catabolic pathway: stage 1, stage 2, and stage 3. Now, there is another second currency that the cells use, and this is your NADPH plus H. So as you can see, through the degradation of complex metabolites, in the presence of ADP and inorganic phosphate, okay, there is also synthesis of ATP and a second currency, which is your NADPH plus H. And this NADPH plus H is usually used for reductive biosynthetic reactions. A good example would be synthesis of fatty acids as well as synthesis of cholesterol. Now, what happens to your complex metabolites? Upon degradation, naturally they are converted to simple products, which eventually will be reutilized by the cells to synthesize complex molecules with the help again of ATP, liberating that energy needed to create bonds, and as well as your source of electrons and protons, your NADPH plus H. No? Now, this shows us the relationship between your energy generating as well as your energy utilizing pathways. Now, there are your catabolic pathways, which are energy generating. And as you can see, as these are utilized, this energy produced in the form of ATP are subsequently utilized by anabolic pathways. And as you can see, they try to intersect at this point, okay, whereby at any point in time, the energy charge of any cell would be around 0.85. It's never 1 class, no? It's usually around 0.85. That's the useful energy charge of your cell. So here is your coupling reaction. Ayan. So the first one is your glucose is phosphorylated, okay, with the help of that inorganic phosphate to become glucose 6-phosphate, and water. And then the other reaction is the hydrolysis of ATP, okay, to ADP and inorganic phosphate. Now, the first reaction, the first half reaction, requires input of positive 13.8 kilojoules per mole, no? While the hydrolysis of ATP to ADP in inorganic phosphate liberates negative 30.5 kilojoules per mole. So again, if you try to look at the net reaction, this inorganic phosphate, by the way, let me get my pointer. This will cancel out, no? As well as these water molecules will cancel out. What will be left would be ATP plus glucose would give rise to ADP plus glucose 6-phosphate. And if you look at the algebraic sum of these standard free energies, then you still have a good margin of negative 16.7 kilojoules per mole. No? There. And if you look theoretically, that would still be enough to synthesize another mole of glucose 6 phosphate, isn't it, class? Yes?